So let's begin talking about the structure and function of macromolecules. Specifically today, let's start talking about proteins. Proteins are more than 50% of the dry mass of most cells. Most interactions among cells, between cells, and within cells are the direct result of the activity of proteins. So when we talk about protein function, we include things like structural support, storage, transport, cellular communications, movement, and defense against foreign substances. That is, organismal and cellular support. So here's an overview of protein functions. You're expected to know these and in general know what they do. So for example, we have enzymatic proteins. Again, enzymes are biological catalysts, so they're going to speed up reactions. Probably the best or most recognized example of an enzymatic catalyst is our digestive enzymes. For structure, they're often used for support. We'll talk a little bit later, and I think we've talked already about silk fibers and their role in, and the protein's role in silk fibers and how strong it makes it. In your own body, you might think of collagen and elastin, the connective tissues that keep your body together. Keratin in your nails, and if you doubt that keratin's very strong, realize that horse hooves are made up of keratin. And if you've ever been stepped on by a horse, I can promise you that it's a pretty strong fiber. Your hair is made up of keratin. Feathers are made of keratin. The top layer of your skin, which keeps the water inside your body, is keratinized. So there's a significant role for keratin in terms of structural support, but we also have other proteins like actin that in the cell play very strong structural roles. They also store amino acids. Case in point, we've already talked about albumin and egg whites. We've talked about albumin in your bloodstream. All of these play a huge role in structure, in protein structure. I'm sorry, protein storage, so that they're available when we need them. Casein, which is the protein in milk, is available for the calf to consume and use in their own body. And think about storage proteins and plant seeds. When we talk about protein-rich foods, we talk about things like beans and such that have a lot of protein in them, and those proteins are storage proteins. Transport proteins, which are used to move other things around, probably the best known, and probably, I'm pretty sure they are the most abundant in your body, are the transport proteins of hemoglobin, which transport blood throughout your body. And we'll talk a little bit later when we talk about protein structure. We'll talk a little bit later about the importance of the structure of hemoglobin in transporting that oxygen. Another role for proteins are hormonal. Hormones are proteins that act at a distance from the site of origin. So, for example, we have insulin that's produced in our pancreas, but it's secreted into our bloodstream, and it's active in all the cells of our body to help sequester that sugar that's in our bloodstream. We also have receptor proteins that are located in the cell membrane, especially in excitable cells. And these receptor proteins are responsible for receiving a signal that we call a ligand and bringing about some change inside of that cell. There are contractile and motor proteins like actin and myosin, which are active in contracting our muscles. And they're also active in the cilia and flagella of cells that are used to propel the cell throughout its environment. And then we have our antibodies, which are defensive proteins, which protect against, and I don't like the word disease, but I do like to say that they are responsible for protecting the body from foreign invaders. Not necessarily just pathogens, but other things. they'll respond. You get a splinter, and you'll see that swelling, and there's proteins in there that are breaking down those wood fibers. So it's more than just antibodies. It's any defensive protein. So again, just a reminder, an enzyme is a type of protein that acts as a catalyst, and catalysts speed up chemical reactions without themselves being consumed in the reaction. They may participate in the reaction, but they're not used up. And because of that, they can be used repeatedly so that we don't have to always be making new proteins, new enzymes. Now, that doesn't mean that we don't make new ones, because eventually they are degraded. We have catalytic enzymes that actually break down other enzymes. So we do have to every now and then make new ones. But the good thing about an enzyme is that it can be used over and over and over again, thus decreasing the metabolic load on our body. Ooh, is that the wrong one? I'm going backwards. Sorry, folks. All right, so we've already talked about how enzymes work. It's important that you recognize that the substrate is the substance that the enzyme works on, and the enzyme has an active site where the substrate will bind. In this case, this enzyme is a catalytic enzyme because it's breaking apart the sucrose. The enzyme sucrase is breaking apart the sucrose into individual glucose and fructose molecules. In the process, and you should remember from previous lectures, that is accomplished by hydrolysis, which is using water to split apart molecules. So, enzymes, sometimes you'll hear enzymes. Proteins, you'll also sometimes hear us talk about them as polypeptides. A polypeptide is a polymer built from amino acids, and typically we call things polypeptides until they're about 100 peptides long, and at that point we start calling them proteins. So you'll hear very often us talk about polypeptides that are, you'll hear me talk about a pentapeptide or a hexapeptide or a multiple, a 20 amino acid polypeptide. But generally accepted in the molecular biology world, in the cell biology world, something over 100 peptides is what we recognize as a protein. Up to that, we typically call it a polypeptide. Proteins consists of one or more polypeptides, and they may be put together as a result of metabolic activity, or they may be built in a single sequence. Just as a refresher, amino acids are organic molecules that have carboxyl and amino groups, and I'll show you again a depiction of that, what that amino acid looks like. It's important that you recognize the R group. We'll talk about that in much more detail a little bit later on. Amino acids will differ in their properties due to differing side chains, which again we call R groups, and we'll talk about how those R groups are affected. So if we look here at this illustration of an amino acid, you notice that there is the amino group, colored there in fuchsia. It's an amine group. It's got a nitrogen and hydrogens attached to it. The carbon there in pink, that is our central carbon. The R group, remember R stands for the remainder of the molecule, that's the actual organic group that determines the characteristics of that amino acid. So we have the amino group, the central carbon, the R group, and at the other end we have the carboxyl group, which will lose that hydrogen quite often, and as it does, it becomes carboxylic acid. So, we'll talk about this, the reason that it's called amino acids, right? So it's got an amine group and it's got a carboxylic acid, so we'll refer to it as amino acid. Again, want to stress the R group is what confers the properties to that amino acid. Now, I'm not asking you to memorize these, but I do want you to recognize that there are 9 nonpolar amino acids. Compare that to the 6 polar amino acids. So there are a lot more nonpolar than polar amino acids. Now, may seem odd because we recognize that we are a water-based organism. So the question then comes, why so many nonpolar molecules when we are a water-based organism? Well, that doesn't necessarily mean that those nonpolar molecules are exposed to the polar environment. They actually may be used to determine final conformation based upon being hidden away from the polar environment. Now, in addition to these polar and nonpolar amino acids, we also have some amino acids that are electrically charged. And what that means is we have an acidic and a basic environment. So real quickly, I want you to take a look at glutamic acid, right? So, and you notice here that it is an acid, not only just an amino acid. So here I've got the amine group that's charged, and I've got the acid groups that's charged. But over here on my side chain, so since every amino acid has the same basic structure, the amino group and the carboxyl group, remember it's the R group that makes the difference. So when I look at this group, look at glutamic acid. Notice that there are two carbon chains here. They're all saturated, and then at the end, now this is the R group, and at the end of that R group, I've got this carboxyl group. Now go back here one, and you see the polar group. I have glutamine, right? So here I've got glutamic acid, right? Go over here and I look at glutamine. Everything's the same here, except instead of having that carboxyl group, I've got an amine group on that central carbon. 2 carbons on the chain, the 3rd carbon is bound here to the amine group. So very similar in structure, but in terms of activity, it's hugely different. You know, this is polar, polar capable of making hydrogen bonds, but this guy is acidic. It's an acidic group, which means it's going to steal where it can hydrogen, right? So hugely different, and hugely different, right? But if I look at these and the structures, I want you to notice something else about the charged groups. Typically, they tend to be either basic or acidic, and they tend to be charged further away from the molecule, from the peptide, from the central carbon. And we'll talk about why that's hugely important a little bit later on. Okay, so let's talk about amino acids for just a little bit. Amino acids are linked by our peptide bonds. We've already talked about that. Peptide bonds, again, have the nitrogen from the amine group of one amino acid that's coupled to the carboxyl group on the other amino acid by dehydration reaction. So we have that peptide bond, right? Polymers are linked via peptide bonds all along their way. Doesn't involve bonding between the side chains. So again, we can have a peptide from a few to more than 1,000 monomers. One of the most important pieces of work that's been done in the last few years in terms of neurobiology involves a substance called neuropeptide Y, which is only 5 amino acids long. So it's not about the size, it's about how they work. Each polypeptide has a unique linear sequence of amino acids that we call its primary structure. That makes a huge difference. Okay, so just to go over again a little bit more about structure, we have our peptide bonds. Again, the carboxyl group loses the hydroxide. Amine group loses one of the hydrogens. So when we do that, we release water. It's a dehydration synthesis or a condensation reaction. And when we do that, we form this peptide bonds. Okay. Notice that the backbone stays the same. It's the same sequence. The thing that varies, which makes the protein specific for its activity, is these side chains, or these side chains, excuse me. That's what makes the difference in terms of activity. Okay, so a protein, a functional protein consists of one or more polypeptides that are twisted, folded, and coiled into unique shape. It is this three-dimensional shape that's important. Remember we talked about denaturation. If we destroy that three-dimensional shape, if we change that three-dimensional shape, that means that protein becomes denatured or is non-functional, excuse me. So the functional protein has its three-dimensional shape intact. So if you look here, this is a ribbon model of lysosome. This is what the lysosome looks like in terms of its shape and the distribution of amino acids. If you look at this guy, this is a space-filling model. So if you compare, this is what the model looks like in terms of just the sequence of peptides. This is what it looks like in terms of all their interactions and how much space it actually takes up. So you notice that here in this ribbon model, there's this groove, right? And this is the groove where something fits in. It may be the substrate, it may be another part of the molecule, but these grooves determine how the activity works. If you look at the 3D shape, that groove is very much easier seen, right? So if you look in here, you can see that everything else is occupied. But right here is this little groove that allows it to have its activity, either in terms of bringing about some change or some metabolic activity. Okay, so the sequence of amino acids will determine a protein's three-dimensional structure. The sequence of, alanine, phenylalanine, alanine, lysine, whatever, that determines the three-dimensional structure simply because of those R groups, right? And that structure that's determined by how those R groups interact determines the function of that polypeptide. You will hear me say it, and I know I've said this several times. Form follows function. So the shape of that thing determines how that thing works, or if it works. So here we can see how this thing really works. Here's a protein. Here's an antibody protein. If you notice the shape here, we got the ribbon model. We've got a little bit of a space-filling model there with the dense blue and yellows. And you see how if you look at the flu virus protein, now this is an antibody. What antibodies do is they mark that flu virus protein. They mark it for some cell to come along and destroy it. That's all an antibody does. It doesn't actually destroy anything. Everybody confuses antibodies with antibiotics. But the antibody protein just marks this invading protein for destruction. And it does that by assuring a perfect match, a perfect fit. And that's what vaccines do. They prime our cells to recognize this flu virus portion. They prime our cells to recognize this portion of the flu virus so that it can be marked for destruction. So tomorrow we'll talk some more about the structure and function of macromolecules.